Okay, so good evening and welcome to this special presentation hosted by the Calvert Marine Museum Fossil Club. Dr. Dale Greenwalt, who is our guest this evening, gave this presentation in person at the Marine Museum on Monday, and we're happy that he's willing to give this to us again here Thursday evening. So our guest is Dr. Dale Greenwalt. He's a resident research associate in the Department of Paleobiology at the Smithsonian Institution in Washington. And he has been affiliated with them since 2007, and I believe all of that time in retirement. So his work centers on the description of the amazing fossil insect fauna in the Middle Eocene Keshenin Formation in northwestern Montana. You may recall, some of you would have been present at the presentation that he gave to our Fossil Club a few years ago in November at our meeting at Scientist Cliffs. He is also interested in defining the biological and geological processes that result in the preservation of these insects and the original biomolecular components that are found in the fossils of the Keshenin Formation. So tonight he's going to speak to us on the fossil record of blood. I just love this presentation, so I hope you enjoy it as much as I do. Dale, take it away.
Thank you, Stephen. Thank you very much for inviting me. It's a pleasure to be here tonight. And again, the title, The Fossil Record of Blood, is a surprising one. I don't think very many of us would assume that blood per se would have much of a fossil record. Before we get too far into the seminar, let's define exactly what a fossil is. And let's see, it's not letting me progress from one.
No.
There we go. Sorry about that. Okay, so let's define exactly what a fossil is. And obviously, I'm sure that some of you know the answer to this better than I do. But I think we would all agree that a dinosaur bone is a fossil. We would all agree that a piece of amber with an insect in it is a fossil, despite the fact that these first two that I mentioned here are very different in almost every nature, every aspect of what they are. When we get to something, however, like a 40,000-year-old mammoth, not so much. So here we get into something that we're going to define as shallow time, as opposed to deep time. And let's define shallow time, and I don't think we have to have a definitive definition here, but something less than 1 million years old. An Incan mummy would certainly be something that is, in our definition tonight, a fossil, a shallow time fossil. Let's also talk about ancient biomolecules and shallow time and deep time. Small molecules like, for example, pigments can exist literally for billions of years in the fossil record. There is a 200 million year old crinoid at the Natural History Museum that is beautiful purple in color. And it's due to the presence of a purple pigment that's identical to a pigment that exists in living. Proteins, however, don't last nearly as long. And proteins are a little controversial as far as the fossil record is concerned. And actually, quite a few things that we're going to be talking about today are controversial. It's one of the things that's going to make it more interesting, I think. Most people think that proteins can exist in a sequenceable form, that is intact, for no more than a few million years. Proteins have been sequenced from bones that are about 4 million years old. Of course, Mary Schweitzer's lab has reported protein sequences from dinosaurs that are more than 100 million years old. And of course, that is very controversial. I think everyone hopes that Mary is right, But right now, it's a pretty big question mark. DNA is even more labile than proteins, although it's been isolated from bones about 2 million years old. Let's start at the shallowest of time. About 20 years ago, a scientist in Spain took some blood and he smeared it on a rock and he put it on a shelf in his lab. And 10 years later, he came back and he looked at the rock under a scanning electron microscope. And this is what he saw. Essentially mummified erythrocytes, or red blood cells. These are not fossils, of course, but they certainly contain preserved biomolecules like hemoglobin. And so really, this is at one extreme end of our trip through the fossil record of blood this evening. As we progress, we can get all the way into fossils of red blood cells that are hundreds of millions of years old. These are found in a ichthyosaur. The red blood cells on the left, of course, are totally mineralized. They've been replaced by minerals 100%. There are no ancient biomolecules here. This, however, is very controversial, primarily because the authors here also reported the identification of white blood cells on the far right. But white blood cells are easily one or two orders of magnitude larger than what they have here. And so again, a lot of what we're going to talk about tonight is very controversial. It spans a couple of 100 million years, and I hope you enjoy our trip through it. Anthropologists would love to know if the Neanderthals and early humans, other species of humans, hunted mastodons. We know they hunted a lot of different animals. What animals did they hunt? Well, a lot of museums have arrowheads and spear points and knife blades made of stone that have residues of blood on them. And that blood can be sequenced, and it can tell the anthropologist what prey the ancient humans were living on. This goes back a good 40 years or so, when Tom Loy published a paper in the very prestigious journal Science, where he had a rock, an obsidian flake, actually, that purportedly had stains of blood on it. And here he has a micrograph of that blood. And at higher magnification, he reported the presence of red blood cells on that obsidian rock flake. That data was actually very, very controversial, and in fact was proven to be false eventually. But we've progressed a lot over the last 40 years, and to the point where we have Ötzi. And you've all probably heard of Ötzi, a modern human that was attacked and killed while he was traversing the Italian Alps. His mummy was found by two mountain climbers, actually, seen in this picture. And Ötzi died after an arrow struck him in the shoulder and severed an artery, and he bled to death. And he was quickly covered by snow, eventually 5,000 years of snow, and his body became mummified. You can still go there today. You see in the bottom left of this picture, you can see people hiking through the snow to a couple of buildings at the bottom of the mountain there, where you can stay overnight and you can hike up to the Alps, and you can actually go to the site where Ötzi was found. These are erythrocytes, red blood cells that were found in Ötzi. There's been a huge amount of work by medical folks on Ötzi's condition. But Ötzi isn't the only mummified mammal from shallow time. In the Siberian permafrost, there is large numbers. There are large numbers of mammals that are thawing from the permafrost. Here's a baby mammoth. This too is a mammoth, although it doesn't quite look like one, but the trunk is in the upper left, and we can't see it all that well. Here's a better picture. And when the scientists who discovered this fossil, if you will, 40,000-year-old mammoth, and when they cut the skin off of this mammoth, this is what they found here on the right. It actually looks like a piece of meat. I'm kind of surprised they didn't cut a piece off and try to cook it and eat it. But from that tissue, they can get something that it kind of looks like blood, and they called it blood, but really it's just the liquids that they could squeeze out of that tissue. But in that quote unquote blood, they were able to sequence a number of proteins. In fact, scientists have been able to sequence proteins from mammoths and from Pleistocene horses, as well as Ötzi. And these are a list of all the different blood proteins that they have been able to get sequence information from. Interestingly, or at least to me, interestingly, the proteins here in blood are coagulation factors. That is, they are active in the formation of blood clots. And yet, normally in our blood, they're present at very, very, very low concentrations. Most of these other proteins that have been sequenced, like hemoglobin and albumin and immunoglobulin, are present at very high concentrations. So the question here is, why were these clotting proteins preserved? And perhaps, this is a theory, when they formed a clot, they became more stable and more likely to be preserved. This is something that has yet to be tested, but I think it's an interesting hypothesis. We know that mammoths lived in very cold environments, and they had to keep themselves warm, not only their internal organs, but their peripheral tissues as well. When we walk around the cold, you probably, if you've done so, your fingers have gotten cold, your toes have gotten cold. That's because in response to the cold, our body restricts the blood flow through peripheral tissues. And there's another interesting biochemical phenomenon that takes place, and that is that our blood is very poor at releasing oxygen when it's cold. It binds oxygen and releases it at warm temperatures, but at cold temperatures, it holds on to it and it circulates until it gets to someplace warm, and then it gives up the oxygen. But for a mammoth that lives pretty much full time at really cold temperatures, it has a lot, it has tiny ears to protect them from freezing, but it has hooves and the trunk and whatnot. And some scientists ask a very interesting question about the mammoth. They wondered if mammoth hemoglobin releases oxygen in the cold more efficiently than it does in humans or in today's, for example, African elephants. And so they were able to obtain mammoth hemoglobin DNA. And from that DNA, they predicted the sequence, and they were able to synthesize mammoth hemoglobin in the laboratory. And what they found was that mammoth hemoglobin differed in a number of different places. It had mutations in it relative to, say, African elephants or Indian elephants. And this changed the properties of the hemoglobin. And the scientists were actually able, because they had mammoth hemoglobin in the laboratory, they were able to test it. And what they found was, lo and behold, indeed, mammoth hemoglobin doesn't hold on to oxygen as tightly as our hemoglobin does. And it releases the oxygen in cold temperatures, which makes perfect sense. This is one example of what scientists can do when they are able to isolate and purify ancient biomolecules from organisms that are tens of thousands, even millions of years old, because we can figure out what their metabolism was like back then. And that can tell us a huge amount about how these organisms live. Okay, let's transition now from shallow time to deep time. This is a fossil of a frog found in northeastern Spain. And when the scientists took one of the legs and they made a cross section of that leg bone, this is what they found in the upper right here. Bone marrow. It actually looks like bone marrow. It's red. Hematopoietic bone marrow. That is, this is the marrow in the bone that makes red blood cells, preserved after 10 million years. This is another example, even older. This is a louse on the left, a fossil louse. It's not even an amber, it's a compression fossil. And if you follow my cursor here, right over here, you see this little red portion of this fossil. Well, the scientists compared the fossil louse to a louse that lives today, and the living lice have a stomach like this. And of course, many lice live by sucking blood, and the stomach can become full of blood. And the scientists concluded that, in fact, what they had here was the fossil of a louse after a blood meal. Unfortunately, they didn't try to determine if there were any ancient blood molecules in that specimen. Here's another specimen. Now, it's tough to characterize the fossil record of blood, so sometimes we have to use proxies. That is, we don't have direct evidence for the presence of blood, but we have indirect evidence. This is a tick. It looks like it's bloated. It looks like it should be full of blood. Scientists looked at it with an electron microscope, and they found red blood cells in there. But even more interestingly, they found that some of the red blood cells had parasites, blood parasites in them. And that's important because in the next few examples we use, we're going to be using blood parasites as a proxy. we're going to be using them as evidence for the presence of fossil blood. This is not a mosquito, it's a fly, but it feeds on blood. And living species of this particular fly carry the parasites called trypanosomes. And when they looked at the proboscis of this particular fossil fly, now this is an amber, they found trypanosome parasites. And of course, the proboscis is involved in the transfer of blood from the host to the stomach of the fly. And so you'd expect to find them smeared on the proboscis of the fly. Probably the most well-known insect that feeds on blood, of course, is the mosquito. I don't know how many of you have ever let a mosquito bite you, and instead of swatting it, you just look at it, you watch it, you observe it. What happens is that the female mosquito is after protein. She needs protein to make her eggs and to mature those eggs. And so when she's feeding on blood, she doesn't want our plasma, she wants our red blood cells because they're packed with hemoglobin, the protein that she needs for her eggs. And so they actually excrete the liquid portion of the blood and they retain the red blood cells. This is a mosquito in amber. You can see the proboscis here. And the species name of this particular mosquito is malariager, and that gives you a hint as to the type of parasite that it carries. And when they looked at the stomach of this particular mosquito, indeed, they found malarial parasites. So again, this is a proxy for the presence of blood in fossils that are tens of millions of years old. We're going to skip back just for one slide to shallow time. People sampled tissue from King Tut, an Egyptian, in fact, they sampled a whole series of different mummies. And King Tut here on the left, when they tested for the presence of malarial DNA, it was positive. In fact, they found 2 different variants of malarial DNA, proving that King Tut, over the course of his life, his rather short life, had been infected twice. He had malaria 2 different times. And in the center here, Thuya and Yuya were his grandmother and grandfather. And they too had been infected with malaria. In fact, it's probably safe to say that a very high percentage of the people who lived along the Nile at one point in their life were infected with malaria. Again, this is a beautiful example of what we can do when we can isolate ancient biomolecules, DNA, in this case, from fossils. This is a fossil mosquito, and the abdomen looks bloated and dark, and in fact, it is engorged with blood. This is another one. This is yet another one. And in fact, there's a place in northwestern Montana where we have collected, I think, 7 or 8 different blood-engorged mosquito fossils. It's the only place in the world where they have been found. And the abdomen in this particular one was analyzed for the presence of blood. We didn't look for DNA or proteins, but we did look for, let me go back here, iron. You remember that hemoglobin contains iron. In fact, iron is responsible and needed for the binding of oxygen. And the portion of iron that binds, the portion of hemoglobin that has iron is called heme. And here you have the chemical structure of heme with iron in the middle. And it is this iron molecule that binds oxygen. And so if you have a lot of hemoglobin present, you're going to have a lot of heme. And if you have a lot of heme, you're going to have a lot of iron. And this is, in fact, exactly what we found. In the female abdomen, there was almost 10 times as much iron as there was in the rock matrix or other parts of the female or male mosquitoes, which we were able to collect as well. And of course, male mosquitoes do not feed on blood, so they're good negative control. So knowing that we had iron, we asked the question, well, might we have this intact heme molecule? And so when we analyzed it using mass spectrometry, up on top here in A, this is the spectrum, the mass spectrum of the abdomen of the mosquito. And right below it in B is the mass spectrum of blood, pig blood that we got as a positive control. And it's an exact match. In C at the bottom, that's what we were, that's the mass spectrum of the material in the rock matrix adjacent to the mosquito. No heme there, but there was heme in the fossil mosquitoes, preserved after 46 million years. I had an occasion to travel to the Dominican Republic. Most of you have seen the original Jurassic Park movie, and you probably remember in the beginning of the movie when the lawyer took a trip to the amber mines, and they actually went to the Dominican Republic. Of course, the amber from the Dominican Republic is only about 15 million years old, but they had poetic license when they made the movie, I guess. But this is a typical amber mine in the Dominican Republic today. It's nothing like the beautiful mine that was portrayed in the movie, where they had supporting timbers and electricity and all kinds of equipment. The folks here just simply dig down to a layer of clay where they find amber. In this particular photograph, you see the rope at the top of the picture. It goes down to a pulley and it's attached to a motorcycle engine. And men grab the end of the rope and they're lowered into this mine pit that you see at the forefront. This goes down 100 feet, believe it or not. And in this case, they were able to reinforce the sides of the pit. I guess they don't have OSHA in the Dominican Republic. This is very dangerous, and the men work very hard. They come up absolutely saturated in sweat and mud, and they then sell. Oh, I am not doing this very well. I apologize. They sell pieces of amber to tourists, and they make their living by selling this amber. It's a really tough life. It's a hard way to make a living. So let's progress to dinosaurs. Everyone wants to talk about dinosaurs. So Mary Schweitzer, who gave a seminar, a number of you probably went to her seminar here a couple of years ago. Mary has become very famous by her work on ancient biomolecules and ancient soft tissues in dinosaurs. And she was able to show that if you dissolved away all of the minerals of dinosaur bone, you would be left with this soft tissue. And note the scale by here. It's this this hamburger-like piece of tissue is only a couple of millimeters, maybe, yeah, 2 or 3 millimeters in diameter. And from this nondescript tissue, she was able to show the presence of blood vessels, very tiny blood vessels or capillaries. And she has published results indicating that she has been able to isolate the protein actin from these blood vessels and sequence it. Very controversial. This is a micrograph of red blood cells from dinosaur soft tissue. And she has published work where she has reported sequencing hemoglobin from the soft tissue she's been able to isolate from dinosaur bones. Again, a lot of people who do similar types of science have not been able to repeat this work, but I think everyone hopes that Mary is right. Many people, however, have recently been able to reproduce her work in terms of isolating this soft tissue from bones, this hamburger-like material. Derek Briggs' lab published a very important paper here a couple of years ago where they were able to isolate this material from a number of deep time tissues. And in fact, John Nance from the Calvert Marine Museum has been able to isolate this tissue, the soft hamburger-like tissue. And I refer to it as hamburger because it's reddish in color, no one knows why, from the Ekphora, a 12 million year old snail from Calvert Cliffs. John has not been able so far to find collaborators who are able to characterize the ancient biomolecules in this material. I was able to collect a fish fossil from northwestern Montana, and from that fossil, I was able to pick out some bones. And on the right here, you see this beautiful circular pattern of lacunae, spaces where the cells that make bone reside. And when I took tiny pieces of that bone and dissolved the minerals, even I was able to isolate this hamburger-like soft tissue material. This is a young scientist who, when working in Derek Briggs' lab, did some very important work. Her name is Jasmina Wyman. And what she showed was that this hamburger-like tissue really is made up of proteins and other types of molecules like sugars, for example, that chemically react with one another to the point where they're highly derivatized, highly polymeric, and they're connected with one another, cross-connected to the point where they're really no longer proteins. You can't sequence them anymore, but they're derived from proteins. The chemistry that this is based on is very similar to that, for example, when you barbecue steak, and you get this nice black covering, or when you toast a piece of bread and it darkens, and if you toast it too long, it turns black. The chemical reactions responsible for the blackened surface of steak or burnt toast is exactly the same. It's protein chemically reacting with sugars. The reactive moieties of protein, like amino groups and carboxyl groups, react with chemically reactive groups on sugars, and they form a larger, more complex, highly derivatized, cross-linked, that's the term I was looking for, cross-linked polymeric protein. There are literally thousands of cross-links produced that aren't normally present in protein. And in fact, this process, the chemical reaction is called a Maillard reaction, and it was first characterized by an Afro-American biochemist about 60, 70 years ago. And it's happening in our bodies today. If we have sugars in our blood and if we have proteins in our blood, a very small percentage of them are going to cross-react, and they're going to form cross-links. If we have too much of this going on, it's bad for us. And in fact, many of us have probably seen advertisements on TV about drugs that are being advertised to lower your A1C. A1C actually is a term that refers to hemoglobin that has reacted with sugar, glycated hemoglobin. And of course, we have hemoglobin in our blood, we have sugar in our blood, but if we are diabetic, especially type 2 diabetic, and if we have really high sugar levels, that sugar is going to cross-react with the proteins in your blood, the hemoglobin in your blood, and it is not good for you. There's a whole long list of reasons why high blood sugar is bad for you. But it's that same chemical reaction that's occurring, the cross-linking of proteins to sugar and other molecules, that is making this hamburger-like tissue that we can isolate from bones that are well over 100 million years old. And I can do it in my lab. John has been able to do it in his lab. The question is, what next? Where will the fossil record of blood take us next? Jurassic Park was science fiction 30 years ago, but oftentimes the science fiction of the past eventually turns out to be scientific truth. And maybe 20, 30, 50, 100 years from now, we're going to be able to figure out how to characterize, how to identify the biomolecules that are present in that soft tissue that Mary and many others now can isolate from dinosaur bones. So I want to leave you with the thought that yes, there is a substantial fossil record of blood. The fossil record of other ancient biomolecules is even greater. And the future of ancient biomolecule work, I think, is something that we should all be looking forward to. There's going to be articles in the New York Times about it, and it's fascinating. I can't wait to hear more. Okay, so I will end there, and I will be very happy to answer any questions if Stephen, you can relay them to me. Thank you very much.
Thank you very much, Dale. It was great. Yeah, so if anyone has any questions, you can either put it in the chat or else unmute and just ask him a question. So I was thinking while you were talking, Dale, what do you think would be the next best thing that somebody could discover? Like what would you like to see somebody come up with? Like what if there's a paper in Nature or Science, and if you could sort of fantasize about what that would be, what would you like to see somebody discover?
Well, that's a great question. You know, there's so much work going on now on shallow time material. All of the fossils, the mammoths, the camels, I mean, there's just literally dozens of different species of large mammals that are thawing out of the Siberian and Alaskan permafrost. And both DNA and proteins can be isolated from all those tissues. So there's going to be a huge amount of publications coming out on that work. In addition, there is, of course, the work on the sequencing of the DNA of ancient humans. The Neanderthals and their ancestors. Soon we will have DNA from Homo erectus. And in terms of the amount of effort and money that's being spent now on ancient biomolecules, I would guess about 99% of it is going towards the sequencing of ancient human DNA. And what it is telling us is absolutely mind-boggling. So that is something to pay attention to. You know, 10, 20 years ago, when people were talking about ancient DNA, right after Jurassic Park came out, there were a bunch of reports of the isolation of DNA from insects and amber that eventually proved to be false. And people were saying, no, DNA is too labile. It can only last for 50,000 years max. And then we found DNA that was a little bit older, and a little bit later, we found even older. So as science continues to progress, we keep pushing the oldest age of DNA further and further back. And now it's up to about 2 million years old. Proteins are sequenceable. They can tell us much about the physiology and metabolism of ancient organisms. That now is 4 million years old. I'm looking forward to seeing both of those being pushed back to 5 million years, 10 million years, 20 million years under the right conditions. Hopefully, we may never get to, T-Rex DNA, but hopefully we'll be able to push it back. That's what I'm looking forward to now.
I understand there's quite a sophisticated DNA lab at the Smithsonian at the Garber facility. Can you tell us a little bit about that?
Yes. The anthropology department has a DNA isolation lab set up, and their interests are in plants, the domestication, the history of the domestication of plants. So they're working in shallow time. For example, they're tracing the DNA of corn back 5,000, 10,000, 20,000 years to see, to learn more about how it was domesticated, where it came from. And they're doing that with a number of different crops that originated in South America. And that DNA lab is available to people who want to attempt to isolate DNA from other tissues as well.
Fascinating. We did have a request some years ago here at the Marine Museum for a piece of bone. There was a lab at Harvard that requested it, and we sent them a piece of Sirenian, of sea cow bone, because it's very dense. It's pachyostotic and osteosclerotic, and they did dissolve it, but there was nothing. They didn't find any soft tissue in it, even though John has found soft tissues here preserved along the cliffs. At least in the bone so far, we haven't found any soft tissues. Maybe a periodic or a tympanic bulla might preserve some soft tissues, but so far nothing like that has been found along Calvert Cliffs.
One of the things they found that's very interesting is that the best place to find DNA, there's two best places actually. One is the petrous bone, and that's a tiny bone behind the inner ear, which is very, very, very hard. That's tough to come by, of course, but the other place, the best place to find DNA is in teeth. And so I think the majority of the work on ancient human DNA is done on ancient teeth. It's a great place to find ancient DNA and ancient protein. And so if in the Calvert Cliffs, if you're able to isolate, and I know you are, teeth from different organisms, that would be a good place to start. There's a much better chance of finding ancient DNA in teeth than in, say, long bones.
So if everyone sends me their collection of megalodon teeth, we'll be happy to look in those.
Yes, that would be a very interesting place to look.
We have some beautiful examples of Gomphothere, Miocene elephant teeth, and perhaps that enamel there is very, very dense, very hard. We might find something there.
It's very controversial, of course. There's some paleobiologists who absolutely refuse to let anyone destructively sample, and it is destructive sampling, their specimens. And you know, if you have a one-of-a-kind specimen, I kind of agree with that.
Yes.
But today, sampling them doesn't necessarily mean you have to destroy them, as pictured, in fact, in what we're looking at here. They can with a very fine, small diameter drill into a tooth and harvest some tissue. So yeah, if you have large numbers of really well preserved teeth and you are willing to, quote unquote, sacrifice and destructively sample some of these, I mean, Calvert Cliffs is kind of right on the edge. There's a lot of stuff there that's 10 million years old or so. And who knows, it may be possible to isolate ancient biomolecules from that material.
Thank you, Dale. Does anyone else have any questions or comments before we wrap up? Okay, not seeing any. Dale, thank you very much for this encore presentation. We really appreciate it, and we wish you Godspeed in getting back to the Smithsonian and to your lab, and that the COVID will come to an end. Thank you very much.
You're very welcome. You're very welcome. Same to you. Bye-bye.
Okay, good night, everyone.